speaker is going to be Brian Kraus from Northwestern. He's going to talk about stable variation in human brain network architecture.
All right. Can everyone hear me? Yes? Somebody? Okay, cool. All right. So my name is Brian Kraus. I'm here from Katerina Gratan's lab from Northwestern Department of Psychology. My advisor was supposed to
go closer to the microphone?
Oh, sorry. My advisor was supposed to come give this talk, but unfortunately she couldn't be here. So instead of a really good speaker, you're stuck with a mediocre one. I apologize in advance. I'd also like to say that let's see here. Sorry. There we go. I just defended my PhD the day before I came on this flight, so I just got to add this to my talk. So thank you very much. Unless you're from the Northwestern Office of Sponsored Funds, in which case I did not defend my PhD until after I got back and put in my reimbursement for this trip. So let me jump in though. So my talk is titled Stable Variations in Human Brain Network Architecture and Its Applications to Psychopathology. So my talk outline, I'm going to start by talking about how individual differences in functional connectivity, or FC, are temporally stable and trait-like, and how evidence that these individual differences in FC do not show strong relationships with longitudinal changes in psychopathology. Then I'm going to discuss evidence that these stable trait-like measures of functional connectivity are associated with stable trait-like measures of psychopathology. So the first thing I want to do is talk about how FC is temporally stable and trait-like. So what I'm showing you here are what we call brain networks. So this is a topographic map on the surface here showing you the locations of these different brain networks. These are derived from averaging data together from many different subjects to get a group estimate of where these brain networks are located. And you can see here we have some canonical networks, such as the default mode network in red, the frontal parietal network in yellow, purple we have the singular perculator network, and in black the salience network. And if you compare the spatial layout of these networks here to other group-derived networks, such as the Yo 2011 atlas or the Schaefer atlas, you'll see that in groups of individuals, the locations of these networks are highly replicable, indicating that there's a sort of a group-level structure that's highly replicable across different processing pipelines, clustering methods, et cetera. But then when you go to individual level data, and what I'm showing you here is 8 individuals from the Midnight Stand Club from Evan Gordon's 2017 paper, you'll see that the locations of these networks are actually highly variable in individuals. So for instance, if you look at the dorsolateral prefrontal cortex or in the medial prefrontal cortex of some of these subjects, you'll see that in these highly sampled 8 subjects, there are these idiosyncratic regions of functional connectivity that distinguish them from this group-derived functional connectivity. And the way that we, in our lab, typically distinguish idiosyncratic functional connectivity from group-derived functional connectivity was developed by Ben Seitzman and my advisor, Katerina. So what we have here is a seed map. So this white dot here is what we call the seed. So what I'm showing you here are correlations between this vertex, which is a voxel map to the surface, and every other vertex on the surface here. And because this seed is in a canonical region of the frontal parietal network, there's elevated correlations with the dorsolateral prefrontal cortex and superior parietal cortex, as we typically see in the frontal parietal network. But then when you go to group level data, there can be a lot of variability in exactly where these networks are located. So this is one individual from the Midnight Scan Club, MSC06, and with the same seed here, you can see that we get a very similar pattern to what we get in the group. So there's high correlations in the dorsolateral prefrontal cortex, superior parietal cortex. But if we look at MSC02, for instance, we can see now we get a very different pattern. So now we're getting negative correlations in the dorsolateral prefrontal cortex and superior parietal cortex, indicating that the functional connectivity profile for this individual at this location is not resembling what we typically see when we average data across many individuals. And the way that we quantify this in our lab typically is that we take the values off the brain here and essentially stretch them into a long vector and correlate them. So if we do this between the group and MSC06 of this particular seed, you'll see that this individual has a pretty high correlation to the group in this region because this looks very much like the frontal parietal network in this individual, just as it does in the group. But for MSC02, we get a very low correlation to this region because now what we're looking at here doesn't really resemble the same functional connectivity pattern. And these idiosyncratic regions of functional connectivity are highly stable over time. So what I'm showing you is some work from Ali Duretsky, who is unfortunately now leaving our lab to start her PhD at WashU in the fall. And these black arrows here are showing you these areas of idiosyncratic functional connectivity in 3 individuals. These are 3 healthy adults. 2 of them were scanned 12 years apart. And you can see that these regions of idiosyncratic functional connectivity are highly stable over 12 years, indicating there's a really high stability over time. Or another way of representing this, we have a functional connectivity similarity matrix, and you can see that subject 1, subject 2, and subject 3 are much more similar to each other over time than they are to other subjects, really underlying the high stability functional connectivity. But this is not only just stable over long periods of time, but you can also look at this over shorter periods of time, such as between resting state and task state within the same individual. So here we have 2 maps like I've been showing you here, one derived purely from resting state functional connectivity for the same individual, another one derived from task data. And I'll note that the task data here, we have applied a general linear model to regress out the task activations, and we're using the residuals. This is background connectivity. And if you look at these idiosyncratic regions of functional connectivity, for instance, in the temporo-parietal junction or in the visual cortex here, you'll see that they're very stable across task and rest states, but they're not entirely stable. There are some state-like differences, right? So if you look right here in the medial prefrontal cortex, you'll see at rest, there's this region that is very low similarity to the group. And if you look at the exact same region in task, you'll see that now this actually is pretty high similarity. And when we quantify this a little more statistically here, if we look at within state comparisons, for instance, a split half of task to a split half of task, or a split half of rest to a split half of rest, you can see that these two things are very highly stable. If you then go between states, for instance, if you look between task and rest, you'll see that you get a little bit of a drop off here, so there is a little bit of a state-like effect, but you have much higher stability within subject than you do between subjects. So this is what happens if you compared, for instance, MSCO2 to MSCO3. This is what the stability of these maps would look like. So you do take a little bit of a state hit, but for instance, if you are using task data to try and recapitulate resting state data, you can actually do a pretty good job of it. And overall, these results suggest that functional connectivity is largely trait-like, though there is a smaller state-like component. And how much data do you need to actually do this analysis? So some evidence from Evan Gordon in 2017 and some later papers suggest that it's about 40 minutes. And the way that we get this estimate is that we have a little over an hour of resting state data that we hold out to estimate, and each one of these lines represents one subject in the Midnight Scan Club. So we hold out an hour of data, estimate functional connectivity, then from an unseen split half, we add in 5 minutes, 10 minutes, 15 minutes. And then when we get up to about 40 minutes, you'll see that all the subjects except one are above 0.8. And this subject slept a lot in the scanner and moved their head a lot, and so their functional connectivity estimates were just very poor in general. All right, so now I'm going to talk about some evidence that individual differences in functional connectivity don't show strong relationships with longitudinal changes in psychopathology. So in order to do that, I'm going to have to describe a data set I did my dissertation on. So this is the brain map data set, and this is my collaborators at Northwestern, Robin Nusslock and Rick Zimbarg, who are co-PIs in this grant, along with Michelle Krasky at UCLA. So everybody in this study is 18 or 19 years old when they begin this study at baseline. So at baseline, they undergo a little bit over an hour of fMRI scans, and they also do a myriad of psychopathology assessments. Lots of these are self-report. They also do a structured clinical interview. So they do those same psychopathology assessments at a 1-year follow-up, 2-year follow-up, 3-year follow-up. And then at the 3-year follow-up, they also do the same exact MRI scans that they did at baseline. So in total, we have 202 individuals in this data set that were over our criteria for over 40 minutes of low motion fMRI data to get these reliable individual level estimates. In this section, though, I'm going to be talking about our longitudinal sample, which is a subset of those 202. So these are individuals that had over 40 minutes of fMRI data, both at baseline and at the 3-year follow-up, so we could get separate, reliable estimates at both time points. And there was a good variability of psychopathology in this data set, so all the structured clinical interviews that individuals did at the baseline in each follow-up. These are the diagnoses that were given over the course of past and present for each individual their whole time in the study. So in a total of the 202 participants, we have 79 healthy controls, 35 people that meet criteria for just a depressive disorder, 32 for just an anxiety disorder, and 42 for a comorbid depressive anxiety disorder. And now what I'm showing you are the continuous measures that I'll also be talking about here. So a lot of these are derived from a hierarchical model of psychopathology that Rick Zimbarg developed, who was a co-PI in this grant. And it's very similar to HiTOP if you're familiar with that, but if you're unfamiliar with that, essentially it's a structural equational model with 3 levels. So you have kind of an overall psychopathology factor, and we call that here general distress. Then if you go the next level down, we have 2 transdiagnostic factors, one for depression, which is anhedonia apprehension, another for anxiety, which is fears. And then we have a bunch of narrower factors, which are more specifically looking at just depression or just anxiety symptoms. And our other indicator here is mean clinical severity rating. So when individuals are assessed at the SCID, a clinician has to rate them from 0 to 8 for the severity of symptoms that they have for every single disorder. So we can just take the mean across every single disorder assessed just to get an overall rating of how a clinician rates your overall levels of psychopathology severity. And so what I'm showing you here now is the stability of functional connectivity over 3 years. I know we're mostly trained never to look at the diagonal in a correlation matrix, but that's where the magic is here. And so what I'm showing you on the diagonal is the correlation or the stability of functional connectivity between an individual at baseline to themselves 3 years later. And in the off diagonal, I'm showing you the correlation between their functional connectivity at baseline to everybody else 3 years later. And as you can see by the much hotter colors here on the diagonal, everybody is much more similar to themselves over 3 years than they are to other people, indicating that functional connectivity, again, is highly stable over years of time. But then what we wanted to look at was to see if this stability varied at all according to psychiatric diagnoses. So for instance, if you were diagnosed with a disorder at baseline of the 3-year follow-up, would that affect how stable your functional connectivity was over that time frame? So what we have here are healthy controls in blue and people who had any diagnosis at the baseline or 3-year follow-up here in red. And each one of these dots is representing the test-retest reliability of an individual to themselves at baseline to themselves at 3-year follow-up. And what these black dots are representing down here are kind of permutations. So this is, for instance, this individual's functional connectivity at baseline to every other individual at 3-year follow-up. So we just see how similar people are to themselves versus other people. And just as I showed previously, everyone's much more similar to themselves than they are to everyone else. And here there are no significant differences between stability for healthy controls versus people with any psychiatric diagnosis. But I don't think this is the most interesting question to ask here. I think you could also ask, well, maybe it doesn't matter whether you had a diagnosis, but whether your diagnostic status changed. For instance, maybe you were healthy at baseline and depressed at 3-year follow-up. Maybe you were depressed at baseline and you were diagnosed with anxiety at 3-year follow-up. So we did a different analysis where we grouped individuals by whether they had the same diagnosis across both time points versus whether their diagnosis changed. And we, again, found no significant differences here, indicating that functional connectivity was equally stable for people who had the same diagnosis versus people whose diagnosis changed. And then I wanted to look at the continuous measures of psychopathology I told you about earlier. So here we have different scores that we look at. So each one of these, we have a score for somebody at baseline and a score at 3-year follow-up. So we take the different score between them. We get an absolute value, so we get the magnitude of change across time. And then we look at how that correlates with their stability across time. And here, after you correct for multiple comparisons, here I'm showing you in red the real values. The black values are 10,000 permutations where I shuffle the scores across individuals to get a null distribution. And after multiple correction, none of these are significant, indicating that these dimensional measures were also not associated with how stable your functional connectivity was over time. So now I'm going to talk about how stable trait-like measures of functional connectivity are associated with stable trait-like measures of psychopathology. So having shown that there weren't really differences over time, now I've just taken everyone's data and averaged it together from the baseline and 3-year follow-up to get sort of a general level of how bad your psychopathology was and what your functional connectivity estimates are. And in order to do this, first I'm going to have to describe this metric called network dissimilarity. So what we do here is we start with a group level atlas here. This is the Gordon 3-3 functional atlas. And you can see here we have, for instance, the default mode network represented here in red. And from this atlas, of course, if you pairwise correlate all the time series for these parcels, you can generate a functional connectivity matrix. And if you do that for only the red parcels here in the default mode network, you can get this within network connectivity for the default mode network block, an estimate of this. So I've just zoomed that in right here for subject 1 and subject 2. And then what I did here is I just took the upper triangle of this matrix for subject 1 and subject 2, and I just stretched it out into a long vector, and I correlate it, right? So if subject 1's connectivity profile within the default mode network looks a lot like subject 2, they'll get a pretty high correlation value. If they don't look much like each other, you'll get a pretty low correlation value. And then what I did is I just multiplied those values by -1. So now a higher score means you're more dissimilar to everybody else. And then I did this pairwise for every single subject in the sample, right? So I end up with a 202 by 202 matrix, which tells me how similar or dissimilar every subject's connectivity within each network is to each other. And then I just take the mean of each row. And so that will tell me, for instance, for subject 1, overall, how dissimilar is your default mode network connectivity to everybody else in the sample. And I do this for 4 association networks that are commonly associated with psychopathology in the literature. Default mode network in red, the frontal parietal network in yellow, singular percular network here in purple, and the salience network in black. And when I did this, there was only one significant association with the dimensional measures of psychopathology I mentioned earlier, which is overall mean clinical severity rating, or how bad a clinician rated your overall psychopathology across disorders. And so what I'm showing you again here is the real data in red and these null distribution here, these black dots here. And you can see that the salience is significantly different from the null distribution here, showing that as the connectivity in your salience network becomes more dissimilar from the rest of the sample, your overall levels of psychopathology as rated by a clinician increase. But as I was looking at these plots, even though this was the only one that was significant, I thought I was noticing kind of a general pattern where this sort of trend seemed to be there for the salience network across all the measures I was looking at. So when I plotted it, you're rarely right in these situations, but somehow I was here. And so as you can see here, we have each one of these dots or symbols, I should say, represents one of the 7 continuous measures that I described earlier. So we have mean CSR, general distress, fears, etc. And as you can see here, the overall relationship with dissimilarity in the salience network is much higher across these different measures than it is for the default mode network, frontal parietal network, or singular percular network. And if you test these against each other using paired t-tests and you correct for multiple comparisons, they're significant. And also this is significantly different from 0, suggesting that the pooled estimate across these transdiagnostic measures of psychopathology is associated with aberrant connectivity within your salience network. So in conclusion, individual differences in functional connectivity are temporally stable over 10 years in adults and show high stability between task states. There was no evidence for associations between longitudinal changes in functional connectivity and longitudinal changes in psychopathology measures, but I will say as a caveat, we only have 2 data points here, and to make real definitive statements about this, we would need a lot more data points to really tease out whether that's the case or not. But we did find much larger relationships when we averaged across functional connectivity over time between these trait-like measures of psychopathology and stable measures of functional connectivity. So in conclusion, I'd like to thank everybody in my lab, my advisor Katerina, Zach, Deanna, Hajian, Gretchen, Ashley, Ali, Alexis, Derek, and Nathan, and also my collaborators Robin, Rick, Rod, VJ, and the NIH for funding. And also a shameless plug, my poster is currently in the garbage, but if you want to check out some really cool stuff, go check out my lab mate Deanna's poster later. She's doing really cool individualized stuff on functional connectivity and network segregation in younger and older adults. Thank you.